It's my pleasure to call on Noam Sobel from the Weizmann Institute to tell us about, okay, I have it in Hebrew, you'll tell it in English. Okay, it's over here, right. Fine, okay. Imaging, okay, blood, sweat, and tears. Oh, that's good enough. Are you going to sing?
Unlikely.
Okay, please.
So, thank you very much to the organizers for inviting me. Thank you very much to the Adams family for your support of science in Israel. And congratulations to the ORDs for a brilliant start of your career. Our lab has sort of these two parallel domains that we follow. One of them, in brief, is just figuring out how the olfactory system works. How does our sense of smell work? How does our brain enable us to smell things? And the other line of our work is not necessarily figuring out how the system works per se, but rather learning on how the system influences our behavior in health and disease. And today I'll talk only about work from that second part. I think it's more fit for the style of the event and the hour of the day and so on and so forth, because figuring out how the system works is a highly statistical problem. So you can look at this very typical mammalian interaction here. This might look, reasonable to all of you. And what you see here are two large terrestrial mammals, or one large one and one soon-to-be large one, exchanging information through their nose. And this looks, we all understand this. We all, accept it as a path or an avenue for information exchange, particularly social information between these two large mammals. And today I'm going to share news with you that there's another large mammal that also shares information through our nose, yet we're typically far less aware of this. And I'm sticking to politicians just to not insult anybody too much. Politicians don't really get insulted. And of course, you constantly have to update your slides as you go along. That's a real winner. And here you get two for the price of one. So you might say, that still, if you look at dogs interact, you visibly see them sniffing each other very carefully. Now, mind you, humans are almost instructed to sniff their babies, right? That's totally socially fine. We sniff our loved ones. That's not taboo in any sense. But we typically don't walk up to a total stranger and carefully sniff them. It's not typical human behavior. Or is it? So this is the only non-politician you'll see today. And if you see this amazing young girl who's since grown, she has some ancient knowledge in her that the way to greet this hound is by letting him smell her hand. And we're all familiar with that behavior of sending your hand to the hound to carefully sniff it. And so what is this? How does she know, aside from the fact that her father is an olfaction researcher, how does she know that this is the right way to interact with this animal? And this made us think about this behavior. This is a typical way for human strangers to meet, in fact, across more cultures than typically people are aware of. And we started first just looking at people anecdotally behaving in this way. And I'm going to spoil conferences for everybody now. Because we noticed that very typically, shortly after people shake hands, the hand goes like this and like this and like this. The hand reaches the face somehow. So since we don't want to stay with these types of observations alone, we asked some questions. First, we asked whether this information can at all transfer relevant molecules. So we shook hands with sterile gloves and ran them through an instrument called a GC, or gas chromatograph, and asked what sort of molecules go from one hand to the other in a typical handshake. And the answer is a lot. Okay? Now, this may not surprise physicians if there are any in the room, because in hospitals, physicians are encouraged not to shake hands with patients because the hand that shook the hand of the doctor shakes another hand and transfers pathogens in this way. But we also transfer a lot of molecules that are of interest in social chemosignaling. Squalene here is an example for one, that a brief handshake is enough to transfer from one hand to the other. So having used GCMS to find that this is a possibility, we asked if it's indeed a reality. And for this, we designed an experimental paradigm we call the greet. Now, this is actual data. Of course, all the real data you'll see today is people who consented to their images being seen. And in the typical greet experiment, a participant walks into our lab or is introduced into our lab, brought into our experimental room that has a lot of equipment in it, and told to sit there and wait for the experiment to start, and we'll be right back with them. Unbeknownst to them, at this stage, they're being videoed already. And 3 minutes later, an experimenter walks in and greets them, not the one they saw initially, and says, hello, it's a fixed text. You'll be participating in an experiment we're going to conduct. We're still setting up our equipment in the other room. I'll be right back with you. And this occurs under two conditions, one with a handshake and one without a handshake. The experimenter leaves the room, and then we observe behavior looking for this. And this is real data. Okay? Now, before I show you graph data, I'll just show you a brief movie, because this is one case where you really don't need statistics. No sound, please, on the movie. So here you see the greet event. The experimenter is here. You don't really see him or her, but there, you can see them on the back projection image. This is not the ideal video, but the experimenter is now leaving the room, and immediately both hands go to the nose. By the way, there are 300 of these videos online for anybody who wants to play around with it from our lab. Once again, you see the greet. The experimenter is going through the text, whatever it may be. And that was not coded, actually, because it's above the eyebrow. The coding shows up here. But that was coded. The hand went to the nose right away. And this goes on and on and on. I don't know how much of this we want to see. Some participants only consented to having the video shown without their face. But once again, you'll get an idea of what goes on. The experimenter leaves the room. This is not coded, of course. But very briefly, you will see the behavior being coded right about now. There you go. It takes a few seconds, typically, for the hand to go to the face. This happened right away. It's automatic. This is the sum data. Okay? The effect is utterly overwhelming. The f-value is ostensibly 0. Okay? Comparing handshake to no handshake. After handshake, the shaking hand goes to the nose. We did a huge additional series of experiments showing that this is indeed an olfactory behavior. And so when this is happening, apparently there's awareness to what's going on. And to answer our first question is, do people actually sample each other? Are people sniffing each other in behavior? The answer is a resounding yes. We have a series of additional studies on this line, including a cool one published a few weeks ago in Science Advances by Inbal, but I won't have time to go into that now. Okay, so we're exchanging this information all the time. We're sniffing each other all the time. What sort of information might we be transferring, and through what bodily media, right? Because here, I'm going to allude to what Winston Churchill suggested of looking at three potential bodily medias, right? Blood, sweat, and tears. And indeed, all of them are interested in the context of human social chemosignaling. Although blood is very difficult to study for various reasons, sweat we do a lot of work on, and I'll talk about it a bit later on. But I'm going to start off with the curious one, which is tears. Okay, emotional tearing is a curious behavior, right? It was actually specifically pointed out by Darwin as some sort of complete anomaly. Darwin wrote a book that till this day remains the most influential book on the study of human emotional expression. It's called On the Showing of the Emotions in Man and Animals. And in the book, Darwin breaks down every emotional expression to its functional antecedents. Okay, a typical obvious example would be showing of the teeth as a sign of aggression. Okay, so his argument was that this started off from biting, right? The teeth served aggression, and through evolution became a sign of aggression. Okay, a very beautiful set of examples comes from studies by Adam Anderson at Cornell regarding the facial expression of disgust. So apparently the facial expression of disgust is activating the musculature of spitting out from your mouth. Okay, like the word in Latin, disgousia, distaste, right? Disgust. And you activate the same musculature when you're experiencing moral disgust, right? You're not spitting out anything out of your mouth, but it's the exact same functional antecedents. And then Darwin came to tears, which is clearly an emotional expression. And he was at a loss because it was unclear what might be the function or the functional antecedent of tears. So we hypothesized that tears may be a social chemosignal. And to address this hypothesis, we first harvested tears from donors. And this is a talk unto itself, and if we'll have time in the question and answer section, I'll tell you how we did that. But we harvested tears and exposed naive participants to these tears and measured a bunch of measures. And I'm just going to share with you the highlights of this. And perhaps the most meaningful is this. In this case, when men sniff emotional tears, they have a remarkable drop in testosterone. In fact, doctors tell us that this is the most significant drop in testosterone we can generate in men in this period of time. Okay? What you see is about a 20% drop. Here you see it in bar graph fashion. But when I'll show you data, I don't like showing bar graphs. I try to stay away from it. And what you see here, each square in this case is a person. And this is the unit slope line. So you see all the people, and you see they're falling under, most of them under the unit slope line, which means that in testosterone, they had a drop, in exposure to tears, they had a drop in testosterone, okay? The values were higher at baseline than at tears. So we expose people to tears, they have a drop in testosterone that's really, really rapid, and about 20% in overall level. By the way, one of the things that concerns us here is that nobody would ever replicate this because this is a ton of work. So we were happily surprised when 2 years later, an independent group did the exact same thing, had the exact same result. They show it a bit differently. So here they showed the drop, so it's in negative values, and you can see the change in testosterone in tears versus saline, the same as in our study. So we ask ourselves, what may be the behavioral impact of this drop in testosterone upon exposure to tears? And one of the most interesting ones you can think of course, is influence on the behavior of aggression, which is a key behavior, unfortunately, in man and animals. And so we measured aggression. Now, there's a way to measure aggression. It's called the Taylor aggression paradigm, or TAP. And the TAP follows the following rule. So you expose your participants to your stimuli, be it tears or saline in these very experiments. And then you involve them, you provoke aggression. How do you provoke aggression? They are put in a room in front of a computer monitor and led to believe, effectively, that they're playing against another person in the other room, this game. And the game is a game where on every trial they get a sum of money and they have to agree on how to distribute it between them. Now the other person, of course, is not another person, it's a computer algorithm. And to choose a careful word, the other person is a jerk, right? So basically, the other person doesn't agree to anything fair, right? So let's say they get $100, so the other person will say, okay, you can have 99, or I'll have 99 and you can have 1. And if you don't agree, then the money goes to the garbage. And that's the way the other person behaves. So if you're playing this game, you're getting really mad at this person in the other room, right, who's constantly giving you the bad end of the deal. And you play this game, and after a while it ends. And then you play another game that you're playing supposedly against the same person, as far as you know. And this is a reaction time game, where each one of you has to press a button. And the first to press it can then punish the other person. And how do you punish the other person? You blast them with a loud sound. And you can select how loud you want the sound to be by pressing one of these buttons. And this is unpleasant. I'll give you just a brief sense of this experience. And we'll hope the volume is up now, but not too high. So this is basically what it feels like. It's not fun, okay? So you blast them. And then we can measure how loud the sound you used to blast was, and this serves as a measure of aggression. Okay, the louder you blast them, the more aggressive you are. This is a well-validated paradigm. And we found the following. So here you see 25 participants, all of them with pronounced reduction in aggression upon exposure to tears. Here you see, in bar graph form, here you see all the participants with the unit slope line. And once again, you see they fall under the line. You have a significant reduction in aggression upon exposure to tears. Here, there's an instance we kind of like in lab, if I may say so, in that, as you'll see maybe towards the end, a lot of our work is testing whether all sorts of effects that have been validated in rodents appear in humans as well. This is a case where science took the opposite path. And after we did this, a similar effect was discovered in mice. Okay, so there's a chemosignal in mice tears that reduces aggression. In mice, in this study, they actually found the actual molecule. It was, in fact, a peptide. And you can see that as they increase the level of the peptide, then the aggression dropped in the aggressors. In other words, we're now thinking of tears as a chemical blanket that you sort of put on yourself, protecting yourself against aggression. Okay? Now, of course, it would be nice to see if we can figure out what in the brain is driving or controlling all of this. And in our lab, we use several measures or several techniques to measure human brain activity. One of them is this, where we actually insert an electrode up to about here to the human olfactory epithelium, where we record neural responses in a behaving human. It's the only setup in which you can do that without penetrating the skull. And a second approach is using a method known as functional magnetic resonance imaging, functional brain imaging that you're all likely familiar with, and we've developed methods to generate odors in a controlled manner within the magnet bore. And that's what we'll use today. But I'll stop talking now about the actual original biological tear liquid and concentrate on one specific molecule. The reason to use a specific molecule in this case is because it's just easier to obtain than harvesting emotional tears. And this is a molecule we sort of centered on. It's called hexadecanal. I may call it hex from here on. And hexadecanal was identified by others as a potential cross-mammalian social chemosignal. Now, most chemical signals are species-specific. As you can imagine, that might be important. But some are considered cross-species in mammals, and this is one of them. It's considered a social buffering cue in mice. Social buffering basically makes mice chill in a simple term. And humans express a receptor for this molecule and also emit it from various bodily sources. So we're now using hex. And we went back to the exact same aggression paradigm, hypothesizing that hex may reduce aggression. And as you can see here, this is a result from 60 men. Hex remarkably, and I use this word without hesitation here, remarkably reduced aggression. These are 16 trials throughout an experiment. And you can see that in every trial, hex significantly, almost in every trial, hex significantly reduced aggression. This is the z-test between the two events. And you can see that hex reduces aggression in men. It has no perceived odor, by the way. You sniff it, it has absolutely no odor, and it reduces aggression in men. So at this stage, we said, wow, cool, our hypothesis is panning out, great. And here's the result in women. It went the exact opposite way, okay? Hex increased aggression in women across 60 women in almost every trial. The z-value almost never goes down. This kind of blew us away. This was really odd. Now, I should say in a word, I don't like studying sex differences. You get in trouble. It's murky waters, and I don't really think it exists that much in the brain. But we could not disregard this, right? This is a remarkable dissociation between the sexes in the response to this odorant. We repeated this again to see if it happens again. It happened again. So we put it into the MRI to try and understand the mechanism. By the way, this is just the behavior by gender. And you can see this is almost as powerful as a chromosomal test. I mean, if you look at this figure, the way all the men go above the line and all the women go below the line, except one. I mean, this is, behavior doesn't work like this usually, okay? This is really, really odd. So we put this into the MRI. And we found, first of all, that hex activated a region of the brain that's not an olfactory region per se, but rather a region involved in social appraisal. This is a region called the left angular gyrus, a region involved in social appraisal. And so this social odor was in a way, activated the social brain. I'm just going to look at my clock here for a minute and see how long I've been talking. How am I doing on time? 20 minutes, fine. So we see that hex activates the social brain, not the olfactory brain so much, which is interesting. But it activated it identically in men and women, unlike behavior. That's odd, right? You have an opposite behavior in men and women, and initially you observe the same pattern of brain activity. we're missing something by definition because something is driving the difference in behavior. So we did a further analysis that's referred to as a functional connectivity analysis. What this analysis does is it looks at every region of the brain that the left angular gyrus talks with, as it were, communicates with, okay, and asks how hex affects this communication, or whether it affects communication, okay? And you look at all the possible connections that you can have in your entire data set. And this yielded the following two aspects of results. First, under the condition of hex, under exposure to this molecule that, again, we're emitting from our body, the communication was affected primarily with a network of brain structures that are involved in execution of aggression. One key set region you may have heard of is the amygdala, and there was pronounced connectivity with the amygdala, and also additional ventral temporal regions. So hex impacted the connectivity between this area of the brain that it's involved in social appraisal and this area of the brain that's involved in aggression execution, okay? But now we ask, okay, how is it impacting the connectivity? And here reemerged our utter dissociation by sex, okay? What we see is that hex reduced the connectivity in women and increased the connectivity in men. So this is a rare case in fMRI where we actually get a mechanism. It's hard to get a mechanism often with fMRI, but here we have a mechanism. Because what's happening, right, is in a way this is showing us that you could think of the default brain response as aggression, right, in this case. But what's happening is the left angular gyrus is sort of exerting control, right? It's this social appraisal area. Again, it's dampening the response of the aggression execution. Yet when you expose to hexadecanal, in women this dampening is reduced, so the aggression can go up. And in women, the dampening is increased, so aggression goes down. Okay? So we actually have a mechanism for this sex-specific response to this molecule. And yet, we were stumped by this notion of, and here I should say that the eureka moment here came from the graduate student who led this at the time, Eva Mishor. She's now graduated. Because I was telling Eva, I said, look, what's the logic of this? I mean, what in behavior, where in the world can this play a role? Why would a body odor increase aggression in women and block aggression in men? It makes absolutely no sense to me. And she came back after 2 days or so and said, no, I figured it out. Okay? And what was I figured it out? Well, remember this interaction we saw from the beginning of our talk? This interaction can go in various ways, and unfortunately in all mammals, including sadly in humans, one way it can go is this, okay? Males often aggress against offspring, even their own, let alone the offspring of other males. By contrast, maternal aggression is often protective, okay? So if you're this guy, you have a profound interest in making him less aggressive and making her more aggressive, because both of those will serve you. So there's reason behind this behavioral pattern. And with that in mind, we contacted a group in Japan that has perfected the sampling of the odor of baby heads, okay? So they actually sample body odor from one-day-old babies, from hours after they're born. And we suggested a collaboration to this group and asked them to measure the volatiles of interest and the range of interest from the baby head. And guess what? Hexadecanal is the most abundant semi-volatile from baby heads. Okay? So when you see this typical picture, courtesy of Google Images, right? This little baby is actually conducting chemical warfare here, right? Because she's making him less aggressive and her more aggressive. And both of those serve her well, statistically, sadly, right? Okay, so can we identify the brain mechanism for this? Yes, we did, through modulation of connectivity between aggression subnetworks. And finally, if chemosignaling, social chemosignaling in humans is important, as we think it is, might it be part of social impairment? And of course, there are several instances of social impairment in humans, but perhaps one of the best studies is autism spectrum disorder, which first and foremost is an impairment of social interaction. I mean, obviously, there are lots of other things related to autism, but first and foremost, by definition, it's an impairment of social interaction. And so we set out to ask whether social chemosignaling is impaired or altered in autism spectrum disorder. And here, back to our slide of blood, sweat, and tears, we went back to sweat and used a sample or an entity that's sort of the flavor of the month in social chemosignaling in humans for the past few years. And this is something referred to as the smell of fear. Okay, there's a cultural awareness to this as well, that people, when they're in a state of fear, they emit a particular body odor. There's this cultural notion that dogs are well aware of this and can smell fear. You may be interested to learn that that was actually validated a year ago. So this thing that, I know it since everybody knows it since they're a kid, it was finally actually shown in an experiment. Dogs can smell fear. Other humans can smell fear, we know this. And typical experiments or typical studies of the smell of fear around the world have all sorts of paradigms to induce body odor of fear. The typical classical cases are a statistics exam or engaging a person in a public lecture in front of other people. This raises stress to some extent or fear. We wanted something more visceral, so we throw people out of airplanes. It's very effective to induce fear. We actually don't conduct the experiments in midair. This was just our jumping instructors did this for us to make a nice picture. But we throw people out of airplanes and collect body odor as they're coming down. We have very high fear body odor. We started this off independently. We now have a collaboration for about 3 years with the Israeli Parachuters Brigade, and we collect body odor from every first jumper on their first jump. Which now includes women, which is great. So through the pilot's course. So now we're using fear sweat as a stimulus. And I'm just going to show you one primary response. So this is a measure we call GSR, or galvanic skin response. What we're measuring here is the conductivity of the skin. And as your stress increases and your autonomic arousal increases, then your GSR increases. And this is a classic measure. You might also be familiar from seeing in things like lie detectors that also measure GSR, among other physiological measures. It's a great measure. And here you see a bunch of participants exposed to either fear sweat or control sweat. And control sweat is from the same donors, only from mild physical exercise. And you can see a pronounced GSR response to the smell of fear in these young, typically developed participants. So phrased differently, what you're seeing here is that fear is a bit contagious, right? You smell fear and you become a bit fearful yourself. So this is the response in the typically developed adults. And here's the exact same experiments in high-functioning adults with autism spectrum disorder. It goes the opposite, okay? And this was one example of a set of experiments we conducted on this front. The response to social odors that in most cases have no percept. In this experiment, the odor was hidden within a perfume that covered it up. So there's no perceptual difference between the two stimuli. Nevertheless, you have this pronounced difference in autism spectrum disorder, one that's both related to the extent of autism in the autistic participants, and this is a measure of autism called ADOS, and this is the level of difference in response we saw with our measure. And interestingly, also in our healthy participants, there's a measure called AQ, autism quotient, which you can measure in healthy participants or typically developed participants as well. And in the typically developed participants, we also saw a mild but significant correlation between our measure of the response to the smell of fear and autism quotient. So this is definitely part of social behavior in humans. Now, I've gone pretty fast, and I have one small additional story I can tell you about on this line, or I can end here. What's my official time? I have 10 minutes. And then I'll tell you a 3-minute story just to end with, and then we'll have 7 minutes for questions. So I'll quickly skip my ending slide and go further. This is one more place where human social chemosignaling may play a role in human behavior and health in our view. And this I'll just tell you about because it's an ongoing study in our lab, so you'll know where we're headed as well. the most dramatic effect in mammalian social chemosignaling, hands down, is the Bruce effect. The Bruce effect was discovered by Margaret Bruce in the UK in Oxford in 1959. And this is an effect whereby if you take a pregnant mouse and expose her to the body odor of the non-fathering male or a non-fathering male, she will miscarry the pregnancy. Now, the effect is remarkably robust, and what do I mean by that? 80% of occasions. As Mona will tell you, in biology, 80% is 100%. I mean, if you expose an animal to an odor and she will miscarry in 80% of the cases, that's as extreme as you can get. There's lots of research on this effect in mice. We're positive that's an olfactory effect. How are we positive? First of all, it's enough just to bring the odor of what's referred to in the science jargon as the non-stud male. You don't have to bring the male himself. You just need to bring his body odor. And of course, the most critical set of experiments was that you can ablate a small structure in the female nose and you kill the effect. Okay, so then the pregnancy will not be lost. So it's definitely an olfactory effect, and it's a strong effect. Now, humans have lots and lots of miscarriage. There's huge disagreement in that field on the actual numbers because some physicians and researchers say 90% of all human pregnancies end in miscarriage, right? 90%. Now, that's the number because, of course, in many cases, you would never know if there was conception and a day or 2 days later there was a miscarriage. That would not even be bleeding. You would never know. And so the estimates range from 90% to 15% would be the most further downstream notion, and 30% is a number really everybody agrees on. Beyond that, there are cases where couples experience repeated pregnancy loss, RPL. And within repeated pregnancy loss, there's unexplained repeated pregnancy loss. Okay, so physicians, medicine have no clue on why this is happening. We have the hypothesis that there may be a Bruce-like effect occurring in humans. So some sort of body odor trigger involved in this. Now, to address this hypothesis is, of course, very difficult because you cannot do causal experiments with human miscarriage, and I won't say anything political about the US right now, although it comes to mind as I'm talking. But you cannot do any causal experiments in miscarriage. So the first thing we did was something completely circumstantial, and we just asked whether women who experience repeated pregnancy loss have an altered response to body odor. And in a word, the answer is yes, but I'll show you exactly how. For the Bruce effect to work, the female has to have a memory of the impregnating male's odor, right? Because she has to know that that's not the guy, right? So to know that that's not the guy, she has to know who the guy was, right? So there has to be a memory of the odor, okay? This is a prerequisite for the Bruce effect to work. In mice, we actually know where this memory is maintained in the brain, okay? Here is a behavioral set of experiments with women experience unexplained repeated pregnancy loss, URPL, or control women, on their ability to identify the body odor of their spouse. Now, this may disappoint many people in the room who are convinced that they can easily recognize the body odor of their spouse. No, you likely cannot, okay? And here are the control women, and here are the women who experience RPL. Okay, so consistent with the mechanism of the Bruce effect, women who experience RPL can identify their spouse by body odor, and this is how different they are from control women. Moreover, when exposed to male body odor, they have a pronouncedly different response. Okay, so this is exposure to the body odor of strange men, not their spouse, and you can see, and this is a difference map, and you can see that in the hypothalamus, an area involved in the control of the hormonal response of the body, and in additional brain regions, primarily the insula, the anterior insula, we have a pronounced difference in response to male body odor in women experiencing RPL. So we published this first phase of the work of this difference, which in our view is now indeed enough to do something less only correlational, And we're now actually starting, as we speak, a study where we're physically blocking the nose of women experiencing repeated pregnancy loss in an aim to maintain the pregnancy. This is in collaboration with the Clinic for Repeated Pregnancy Loss in Sheba Hospital. So with that, I'd first like to thank all of you for your attention throughout this presentation. And I'll also say, of course, that really I'm blessed and lucky to have a remarkable group that really just enables me to go around and talk about what they're doing. And of course, we're supported by several bodies without which we would not be able to do all this. So thank you very much, and I'll take it. So we have no longitudinal data on an amygdala response in these studies. And again, actually, the amygdala data I was showing you was from the tear studies. And those are horrible studies to run because it's so hard to obtain the material that there are all these questions that you want to ask. And indeed, when we present that work at conferences, it's immediately, oh, did you do this? Did you do that? And no, we didn't. We can't. Because it's just because collecting the data, I mean, the material, right? Because, so I didn't go into the nuances of this, but in one of our studies, we found that the influence of emotional tears on perceivers works only if you use at least an entire ml of tears. That's a lot, okay? And it has to be flash frozen and thawed in a very special way. It's just a horrible type of experiment to run. So, with
make people cry. Huh?
We did, we did. So it's just, we're left wanting with not answering many questions, and we definitely did not do anything longitudinal on the amygdala response, or any of the responses for that matter.
But can you use hex itself?
So, right, so our discovery of hex is very recent, and that's why we're so happy about it. I mean, Weizmann Institute is happy about it as well because they patented it, but we're happy about discovering hex because it allows us to now, instead of torturing people to obtain our stimulus, we just order it from Sigma Aldrich, and life becomes easier. So we'll be able to start doing all these things and answering these questions. I mean, on the same.
What's your prediction about an age effect?
I have no clue. Yeah.
So no, how about tears of joy? Is there something like this? Do you think they have something different in them?
Yeah. So we tried to generate them and failed. So it's just unlike, for the emotional tears, we had an arduous screening process for obtaining criers, which also generated an unfortunate gender bias in our study, right? Because, I mean, it started off by putting up ads, we're looking for volunteers who cry easily so that we can obtain, tears, and we got about 100 women and one man, right? And it's just, it's a social thing, right? I mean, so even if you do have men that cry, they won't come forward in the experimental setting. And so, and then from those 100, you screen them, and we have a screening process, and you end up with about 3 or 4 who can generate an ML. The numbers are insane on that whole experimental process. So, and then we tried doing tears of happiness or laughter, and we never managed. I mean, people get their eyes watery a bit, but it's not the same thing. I mean, if you would see one of our criers, and I have a video somewhere here, right? It streams, right? It streams because we collect, over an ml per session.
How about onion?
So so no, because there are two, well, there are at least 2 ways to generate tears from your eyes. And onions is a protective response because the certain molecules in the onion activate the trigeminal nerve, and that drives the tears. And believe it or not, the molecular contents of reflexive tears and emotional tears is totally different. Okay. So we can't use those tears. In fact, we can use them as a control, which we're doing in some studies.
Okay, any further questions? So now before you go down, David, I invite you to give Noam a small present. This is yours.
Yeah, so, okay, so what David is asking me about is the work I didn't talk about today. So as I told you, our lab does these two very, very different things. And one of them is to try and figure out how the system works. And there, we've been chasing sort of holy grail for about 20 years. And that is figuring out the rules that link the structure of an odor molecule to its perception, right? So until recently, at least, there was no scientist or perfumer who could look at a molecular mixture and predict for you how it will smell, or smell something and tell you what could or should be its molecular structure, right? In contrast to trivial examples from other sensory systems, let's say color vision, right? We know we can measure the stimulus physically in wavelength, and we know that if it's about 400 nanometers, it'll be blue, and about 700 it'll be red, and anywhere in there in between, right? So, tell me what the structure is physically. I'll tell you what you're going to see, or tell me what you're seeing, and I can tell you what the structure is. There are exceptions, of course, and boundaries, but as a rule, that's the case. And in olfaction, we didn't have that, and we've been chasing it for a long time. And about a year ago, a brilliant joint student of David and mine, Aaron Ravia, solved this problem really in a paper published in Nature, where we demonstrate that we can now predict the odor of any molecular mixture by its structure alone. So beyond setting certain boundaries for how the system works, this may be technically exciting as well, because it provides the foundation for digitizing smell, right, for sending odor over IP. And in fact, 2 weeks ago, I think we did that for the first time ever. So we had a group in Germany using a device that measured an odor, transmitted the information to us over IP. We have a device in our lab that recreated the odor and generated it, and it was the same odor. So, don't hold your breath for this showing up in your cell phone. There's lots of really complicated and painful technical things to solve, but they've become more technical and less theoretical than they used to be.
Great, wow.
So a small gift for you, Noam.
Thank you.
From the Academy. Thanks for the lecture.
Thank you.
Thank you again, Noam.
Thanks.